Right, there we go. So, welcome everyone. This is our fourth webinar in a series that myself and Alpesh Doshi started during the lockdown. We know that some of you are at home, and I'm sure a lot of you are very busy. We've both been very busy. I've been working a lot on our online education for my master's students at University College London, but we wanted to have the opportunity to spend some time talking to you about some of the hot topics in embryology. And the four topics we chose for the webinar actually came from a tutorial we did with our students taking our distance learning certificate in clinical embryology. So this talk is a mix of two things. It's about IVF lab troubleshooting and culture media. And I think the two are very much entwined. I think culture media is one of the main important parts of our lab, obviously, and we have to get that right. And there could be a lot of issues around the culture media not being correct. So Alpesh and I discussed the best way we thought to do today's webinar. So what we decided is I'm going to give you a short talk about culture media, and then we're asking you to give us your questions in the Q&A button. So in the Q&A, please put any questions you have specifically about troubleshooting in the IVF lab. And as always with our webinars, after my talk, I will do a short poll and ask you some questions about your labs to find out a little bit more about you. So without further ado, I'm just going to start by just giving a little revision of where we are with our certificate in clinical embryology. So I've been working as an embryologist back from 1987, and then I joined the Hammersmith Hospital to do pre-implantation genetic diagnosis in 1992. But in 1996, I set up my first, and I believe the world's first, human clinical embryology workshop, where we taught people to do embryo biopsy for PGD. And these workshops have been very popular, and we've done about 50 around the world, with most of them being in the UK. And Alpesh joined me very near the beginning, and we've been a great team. And so quite a few years ago, we set up the Embryology and PGD Academy to make our training for embryologists much more formal. So we've done our hands-on workshops. We do want to do another workshop this year, but it all depends on the COVID situation. And we don't want to do this if people cannot fly to the UK. So we have, I think, all of your emails. So after this webinar, I will be sending you the recording of this webinar. We put it on YouTube, and I will have your emails, and I will let everyone know when we decide to do our next embryo biopsy workshop. But if you go to the Embryology and PGD Academy website, you'll see lots of photos from previous workshops and lots of information about those workshops. I will give you that email, that website, sorry, again at the end of this talk. So a few years ago, over all these years, we've had many people ask us to teach them formally about the theory of embryology. And this is something I was very passionate about back in the 80s when I started embryology. I was aware that we're taught by others in the lab how to do the practical parts, but we're not actually taught the theory of what we do. So Alpesh and I decided we'd set up the distance learning certificate in clinical embryology. And I want to stress, this is all online learning. There's nothing face-to-face. So you can do this wherever you are. And we have people all around the world who are taking this course. Now, there are other courses coming up now that do some training in embryology or examining in embryology. I was one of the founding members of the ESHRE working group that set up the ESHRE certificate. But there's nothing that gives you formal training. So this is really the syllabus. We've given you here the syllabus of what we think clinical embryologists need to know. So this is covered in 8 modules, from the laboratory through to reproductive health. And I'm not going to go through them all again today. There's various videos on our YouTube channel, which I'll give you a link to at the end, which tells you more information about this. So I just want to concentrate on module 4. we're very excited. This is a really key module for clinical embryologists, and it is being launched this Friday. We're just waiting for one more talk to be recorded tomorrow evening, but we really have the world experts here, or some of the world experts in lab design, quality assessment and troubleshooting. So we have Steve Harbottle's going to talk about lab design and construction. Jason's going to talk about managing the IVF lab, really important talk about thinking about staff, etc. Raga talks about the air in the lab and the volatile compounds. Alpesh talks about quality management, which is really crucial. Bashak's going to talk about troubleshooting and especially embryo culture. Her talk really is excellent to complement the short talk I'm going to give today. Dean's done a wonderful talk about monitoring your key performance indicators, which is really essential for embryologists to do. And Ronnie, the king of auditing, has given us a really detailed talk about auditing of the IVF lab. Now, all of these things are things that you may not do. It is possible to run an IVF lab and not do any of these things, but it's really the quality of what you do that's absolutely key. So this is what we want to talk about today, and this is what's we've gone through in this module. So a few years ago, you'll see myself and Alpesh Doshi here with some of the key people who were involved in the Cairo consensus document on IVF laboratory environment. And some of those you'll see are speaking on this module. And obviously we have this webinar today. As I've said, it will be on our YouTube channel, and it will also be uploaded to module 4. Now, on the first 4 modules of the certificate, we have a logbook. Now, it's optional whether you do the logbook or not. All of the modules have an exam, which again is optional, and the logbook for the first 4 modules is also optional. And the logbook is something where you record that you have done a number of these procedures. We're not checking your competency, which is very important in the quality of your laboratory, because that would be very difficult to do remotely. But we are checking that you have at least accomplished some of these procedures. So the logbook for module 4 talks about lab design and takes into account VOC levels, quality management and troubleshooting. So with the certificate, you can do any module at any time, and you don't have to register for the whole certificate. So if you're just interested in learning about quality and troubleshooting, you can just register for this module on Friday. So to apply for the certificate, you go to embryologycertificate.com, and as I've said, you can apply for the full certificate now, and you can pay for that all in one go now if you want, or you can just do individual modules, see how you get on. I'm hopeful that you'll like the modules, and then hopefully do the whole thing. So it's very, very flexible. But we have a special offer that was for April, where you can take one module for just £100. We have extended it because we haven't opened module 4 yet. We've extended it to midnight on Friday. So on Friday, you can register for module 4, use the code 150 off, and you'll get this module for just £100. So let's talk about culture media. So with Nikki, was one of my students several years ago. Back in 2014, we wrote a review in Human Reproduction Update about IVF culture media, past, present, and future. And this really covers some of the things I'm going to say today, but I think it's really important for us embryologists to understand this and to think about all of these, the past, present, and future of culture media. And I was one of the founders of the ESHRE Culture Media Working Group. This is a photo of us here. We had many, many meetings, and I wish I could have shared all of the information that we learned. I think we went for about 4 years. It was a very exciting time. And we started off by having interviews with the culture media companies to find out how they developed media and what they were thinking about in the future. And it really stemmed from 2 things, from the adding of a growth factor, GM-CSF, to the culture media quite a few years ago, which concerned some of us, but also the data from Dumoulin lab, which I'm going to go through at the end of this talk, which found different birth weights of children born using different culture media. And those two things together raised some alarm bells in some of us. And we set up the ESHRE Culture Media Working Group, and we had really some great meetings. So I'm going to share some of that information with you. But we did write one report, Time to Take Culture, Human Embryo Culture Seriously, and it was quite controversial, this paper. We had some people that were against what we said, but we really published this to get people to think about the culture media and what we're using. It's really important to think about it. So just a little bit of basic biology about the development of the embryo. When I started at the Hammersmith Hospital back in the early 90s, I worked in a lab with Kate Hardy and Joe Conahan, and they had been working very closely with Henry Leese of the acquired embryo hypothesis, and they were looking at the metabolic requirements of the embryo, and they did some really groundbreaking work. Up until then, the culture media we had used had just been normal culture media we'd used to culture any cells. And that has some basic salts and some glucose, et cetera, in the basic culture media. But people were thinking that maybe a human embryo is different to a normal cell, and it needs different requirements at different times. And their work concluded that there were different metabolic requirements of the embryo at different stages. So the embryo in our IVF lab is going to be sitting in this pool of culture media where it will take up metabolites, and it's going to use these for its metabolism. And it's also going to excrete waste products that normally it wouldn't be bathed in. It wouldn't be sitting in that culture dish just sitting there. So the embryo has an endogenous pool of metabolites, largely the result of the final stages of oocyte maturation. It's got metabolic turnover of RNA messengers and proteins. From our culture media, it's going to be active uptake of sugars, amino acids, and nucleic acid precursors, and the passive transport, especially of lipids, and incorporation of proteins such as albumin, which combine lipids, peptides, and catecholamines. And Gardner and Wade did a lot of work of this in 2013, and as well as many, many others. That's just one of the references. There's been a lot of work in this area. So what Joe and others, Joe Conahan and others realized was that the embryo pre-embryonic genome activation, and embryonic genome activation is a talk in itself. I'm not going to go into that in this talk, but pre-embryonic genome activation, the conclusion was that the embryo does not require these high concentrations of glucose that we find in standard culture media. There's pyruvate, lactate, and amino acids, which are the primary energy source of the embryo before embryonic genome activation. Oocytes and early embryos generate ATP by aerobic oxidative metabolism of pyruvate, lactate, amino acids, and possibly lipids. So pyruvate can also remove toxic ammonium ions by transamination to aniline. So there was some discussion at that time that maybe the high glucose was actually inhibitory to the embryo and having a very negative effect. But these things change, and we're aware now that a small amount of glucose is not going to harm the embryo as far as we're aware. But after genome activation, the embryo does want glucose. So glucose consumption increases, especially at the blastocyst stage. And glucose is used as a substrate for synthesis of lipids, amino acids, nucleic acids, and also essential for blastocyst hatching. So this really was the foundation of sequential culture media. Around this time, companies decided to make different types of culture media. There was even a fertilization media at the beginning, cleavage media, blastocyst media. Before that, we just used a single step media. And I'm going to come back to this at the end. So let's look a little bit about the history. I think if you know me now, you'll know that I like to look back and see how things get developed. And what I'm going to say in the next few slides is summarized in Nikki and my paper, IVF Culture Media. But remember that with rodents, we can get hundreds, if not thousands of embryos. So we can do really complicated work where we can look at each substrate in the media one at a time and increase it and decrease it and see what happens to the development of the embryo. So back in 1929, Lewis and Gregory worked on rabbit embryos, which they cultured in plasma. We've got people that went on to work in blood clots and all sorts of media. Krebs ringers bicarbonate containing egg white. Who remembers we used to use egg white for freezing back in the 80s? And then we had Witten's cultured mouse embryos in very simple media. Anne McLaren and Biggers got a live birth in the mouse in 1958 using Krebs ringers. And then Biggers and Brinster and Witten worked hugely on different types of media. And who also remembers co-culture, we used to co-culture with different other cells to try and help the embryo develop. So there's been quite a lot of work on the mouse. I stopped at 1971, there's more in the paper. But as I said, in rodents, we can use several embryos, but this is not possible in the human. And this is something I want you to remember. Our culture media that we are used is optimized normally in the mouse embryo. It's not optimized in the human embryo. We would need hundreds of embryos to look at optimization. And we also can't check the end product. The end product is live birth. We can't do that in the human. Well, we can do it in the human. I'll come back to a paper we've just looked at, but we can't do it in an experimental nature. So we have to rely on the work in mouse embryos. But let's have a look at human embryos. So 1944. Rock and Mencken cultured human eggs in serum. And back in the 60s, we see Bob Edwards and Robert Steptoe, who had the first fertilization in vitro using Tyrodes, BSA, penicillin, sodium pyruvate, and bicarbonate. And again, we've got the same teams here, Biggers, etc. And the Australian groups, Moore and Trounsden came in, and then the French group, Yves Menezot, looked at various combinations of different culture media. So when I started embryology in the 80s, we made our own culture media. We spent a lot of time making our own media. And what was really key was a source of serum. I, at that time, I am still a blood donor. So I've had all the checks that you need to donate blood. So we often used to use my serum to culture the IVF embryos in, something that we'd think of unheard of in 2020. But for some of the patients, we use their own serum. So we used to often make individual culture media for every patient, and also freezing media using the patient's own serum. So it's interesting how the role of the embryologist has changed. In the 80s, a lot of our time was making the culture media and the serum. And in the 90s, for me, a lot of time was making embryo biopsy pipettes. So these are things now that have been taken out of our hands. And I'm pleased now that we don't have to do that. So it was really in Denmark that the birth of commercial culture media started. So in the 80s, I was lucky enough to go and visit Medicult. I went with Henning Lundsten, who some of you may know the K system that was developed by Henning Lundsten. He was a great friend of mine, and I spent a couple of trips with him in Denmark, visiting Medicult. And then Cook came on quite early as well. And then we have Cooper, Fertipro, etc. So now these are just some. These are not an exhaustive list of all the culture media. So it's a lot for an embryologist to decide which culture media to use. There's so many different options. So we'll talk about that in our Q&A. So I wanted to now look at the composition of the commercial culture media. And this has been where the controversy lies because of the rights of the companies not wanting to be fully disclosing their culture media. they haven't necessarily been very open in what the composition is. And as a result, we see a lot of differences in the culture media. With the Extra Culture Media Working Group, when we interviewed the companies, they all said they were happy to be fully disclosing their ingredients and the concentrations, but they didn't want to be the first. So they wanted everyone to do this, but we never quite got there. Dean Warbeck, who's on our module 4, giving an excellent talk about KPIs, has led this excellent paper. As they've said in this paper, mainly designed from animal studies, and there's a great variety between the media. I really couldn't believe when I looked into it how different they were. You would have thought that all the media would have been very similar, but they're very, very different. There are strict manufacturing and quality assessment that has to be done with commercial media. And I think that's one improvement on us making the media ourselves. When we're in an IVF lab, if we're making our own media, we really cannot do the quality assessment and these very strict manufacturing to make consistent media. There's so many variables in a lab. It really is a professional thing that has to be done to ensure quality. So they have strict requirements and improved batch to batch consistency and less contamination than we would have done in our own lab. But as I said, we do not know the composition. And how do we monitor the success of a media? Another thing I want you to think about. The ideal way when we've got a new media is to look how successful it is really at live birth rate. That's our holy grail. We might want to look at fertilization rate and check that we're getting good blastocyst development and good pregnancy rates. But really, we have to do huge studies, multi-center studies, looking at media and looking at the success of that media. And I'm going to talk to you at the very end about a piece of work I've done with Daniel Bryson, where we have looked at exactly that in the UK. So how would we do that? Would we do a randomized controlled trial, an RCT? And would we also follow up of the success and the health of the babies born? So these pieces of work that in an ideal world we would do is very tricky to do and would cost a lot of money and push up the price of culture media and put up the price of IVF. So we have to come to a balance of what we know about the media and what we can do in our own lab to check which media we should use. So when we looked at the components of culture media, some of the media are very simple. They contain 12 ingredients, and there are some that contain 78 ingredients. So this is what we had wanted to know from the culture media companies. How did they decide what to put in and what not to put in their culture media? Some of the things that might be in there, well, it needs serum protein source. It absolutely needs that. Hormones, lipids, amino acids, energy substrates, water, obviously, osmolytes, EDTA, vitamins, antibiotics, pH indicator, buffer, growth factors. So how this gets developed is really interesting. And in module 1, I've given an extended version of this talk. And also Kiri Birley gives a talk on sequential and single step media. And she talks about the latest research. So as I said, when we started in IVF, we just had single step media and it was very straightforward. But as I said, from the work of Joe Conahan, Kate Hardy, Henry Leese, etc., people realized that maybe the embryo needs something a bit more complicated. So with single step media, we say it's like a buffet. We give the embryo everything it needs and it decides what it wants to do with those compounds, those reagents. Does it want to take them up or does it want to leave them there? So it's like a buffet. What does it want to pick? And can it pick things at different times of its development? With sequential media, that very much come out of the work of Joe and Kate, with the view that since the embryo has different requirements at different times of its pre-implantation development, maybe it would be better to give the embryo only what it needs at those specific times. So sequential media had lower glucose at the beginning and then more glucose in the blastocyst media. I'm not going to go into the pros and cons. I'm going to ask you in the poll what media you use, whether you use single step or sequential. But the data out there at the moment, as far as I'm aware, is not saying one more in favor of another, which has been quite interesting and shows how the culture media has gone on to develop. The important thing about single step, it became very popular with the use of time lapse. Time lapse is better not to be disturbed. It's easy for the embryologist to put everything in there. So it was really time lapse that gave the surge in the use of single step media. But we can talk about this afterwards. Are we stressing the embryo? So Henry Leese's quiet embryo hypothesis says that a happy embryo should be metabolizing quite quietly. And if the embryo is stressed, then it metabolizes faster. So in the consensus document we did in Egypt, we have gone through all of this. So I don't need to go through all this now, but this will be covered in the various talks in module 4 of the certificate. But all the labs are different. And I said the same in one of the other webinars. If you have the identical setup in identical labs with identical staff, but in different locations, you could have different stress on the embryos and different outcomes. And that's because of the environment of the lab. It's the, what has the walls been painted with? What does is there anything that comes up through the sink, through the plug in your sink, through the waste? What's the air? How's the air coming in? Where is the air coming from? Is it coming from the outside? Is it being recycled from the hospital or clinic you're in? There's so many different things. It's not just the consumables, the equipment, and the people. It's everything. Every lab is different. I did some work many years ago about single and group culture. We couldn't quite come to a consensus on it. Some people felt years ago that it was better for embryos to be cultured in a group. But with PGT and also with time lapse, we've now moved to single culture. But all of these factors, pH, osmolarity, etc., all need to be taken into account when we're thinking about stressing the embryo and when we're troubleshooting in our lab. So these are all key things for troubleshooting. So if you are having any issues with any of these, then please put some questions for myself and Alpesh in the Q&A. Now, I just want to say a bit about CE marking. So when we started the ESHRE Culture Media Working Group, to be honest, none of us were really aware, what does CE marking mean? And by the end of the work, I actually felt it was a useful mark for sure. And in the UK, our authority, the HFEA, says we have to, if possible, use CE marked media. And this was started in 2009 from the European Union, and it regulates ART culture media as a medical device class 3. And it really shows that it's fit for purpose. It shows that the media has been made under good operational standards, and that it's reproducible, and that one batch this year is the same as the next batch the next year. And it's really showing good manufacturing process. What it's not doing is telling you that your culture media is highly successful and it's going to be the best culture media. It's not telling you anything about its performance. It's just telling you about how it's been manufactured and the quality of that. So I think it's useful, but it's not telling you it's fabulous media. And I really want to talk to you about the mouse embryonic assay. We have now become embedded with the mouse embryonic assay. There's no alternative available. People have discussed whether we could use human embryonic stem cells to help develop media, or to be sure that the media is okay. So when a company makes a batch of media, it has to do a mouse embryonic assay. So it has to ensure that that culture media will allow for 80% of mouse embryos reaching the blastocyst stage. So that sounds great, that sounds useful, but there's no standardization. There's no standardization around the strain, and some strains of mouse embryos are very easy to culture to 80% blastocysts, and some are very difficult. It doesn't stipulate the day that the culture has to start, so it could be a short time. It doesn't say the number of embryos, and for cost benefit, most companies do not do this on 100 embryos. They just do it on a handful of embryos. It doesn't talk about oil, proteins, osmolarity, etc. So that's one problem of the mouse embryonic assay. But the second problem is we're growing human embryos. There are some things that are needed for mouse culture that are actually not needed in human culture. But we have to put them in there because the culture media has to pass the mouse embryonic assay. So unfortunately, at the moment, we have no alternative. It's the best we can do. But I wanted you to know about this limitation of our culture media. So I wanted to talk to you now about the Barker hypothesis. So this is Barker here. In 1994, he developed the Barker hypothesis, and he said the birth weight of anyone is linked to an increased risk of early onset of adult diseases, including diabetes, hypertension, and cardiovascular disease. So he said children with low birth weight can have long-term health problems. And he said the physiological response of the embryo to environmental conditions most likely mediated through epigenetic changes that may persist throughout life and into subsequent generations. So basically, how we culture our embryos, not just our birth weight, it's right from the beginning, from culturing those embryos. What we do in just those 3 days or 5, 6 days could have a long-lasting effect on the health of our future children. And this really came to a point with this work by John Demorelin's group, which had showed, this was not the primary aim of the paper, but in this paper, they used 2 different culture media, Vitrolife and Cook, and one of the things they had looked at was the birth weight of the children, and they found a significant difference with Vitrolife giving children of higher weight than Cook. And we weren't sure what's right and what's wrong, or what's compared to normal. That wasn't really the point, and it wasn't really the question. What we really wanted to think about was, what does this mean for IVF? The paper was criticized, and but it really started a big discussion, and they followed this up with the Nielsen paper and looked at the children a few years later, and they've done further work on this since then. And this difference in birth weight does seem to persist. There is a difference in the weight of these children. So this could have an effect if you're thinking about the Barker hypothesis. In the talk, in the IVF module 1, I go into this in a lot more detail, and I go into other factors that could be affected by culture media besides birth weight. We can certainly talk about them during this webinar. These are just some of the papers. There are many, many looking at birth weight. Some say it makes a difference, and some say it doesn't make a difference. I think what you've got to really think about, as I've said, these culture media are very, very different. It depends. If you take 10 culture media, it depends which 2 you compare to whether you may or may not get a significant difference in birth weight. So maybe culture media A and B show a difference in birth weight, but maybe culture media A and C show no difference in birth weight. So this is why we see these contradictory studies, some showing a difference and some showing a no difference. So my take on this is that there are some culture media that have a difference in birth weight if you compare them to another. What's right and what's wrong, I just don't know. And I don't think it will take us years to work this out in the human. So I think these are things we have to be aware of, but I'm not sure this is really pointing that any one culture media is better than another. I think we need to keep monitoring this and do further research for sure. And the Escher Culture Media Group thought about how we could do further research on this, but it is complicated and it would be very expensive. So I want to just point you out, this is almost the end of my talk. I want to point you out here in the top right. There's a paper, as I said, that I did with Daniel Bryson. This came out of Daniel and I work on a couple of the human fertilization and embryology committees together. And we had thought, how could we look at the impact of different embryo culture conditions on AR treatment outcomes? So what we did, Catherine Castillo was Daniel's PhD student. We took 2-year time period, and what we did is we asked all the IVF units to tell us during those time periods which culture media were they using and which culture conditions they were using. So we asked them about incubators, we asked them about number of staff, we asked them many things. There's a lot of quite complicated data in this paper. We had the live birth rate and the live birth weights from the HFEA data, and we crossed these two databases. So, something strange is happening here. Oh, sorry, so it's asking me if it wants to add subtitles. So from this study, as I said, there's a lot of complicated data. One thing we did find, there were significant differences in live birth rate between the culture media. We looked at, there was 8, I think it was 8 culture media in all. So this paper just came out this year. Please read it, have a look, and it's in Human Reproduction Open, so it's freely downloadable, but further work really needs to be done on this, on different data sets. It took us years to do this work, and it was really very complicated. Oh, sorry, my screen's going mad. Oh, I don't know what's happening here. Let me just see what's happened here. Right. Oh, sorry, I'm getting a technical hitch here with my computer. Something is going very, very weird. Right. So that was the end of my talk anyway. Let me just unplug this. I don't know what's happening here. Right, I'll just try and share the screen again. Sorry about that noise. I have no idea what that is. So what we need to do is have full disclosure, composition of the culture media, ideally by the culture media companies. We need more well-designed randomized controlled trials with large epidemiological studies and full transparency. We need reliable standardized models of assessing multiple endpoints, and post-implantation development should replace the mouse embryonic assay. We've got a lot of work to do to get that working. And we need to have structured long-term follow-up of the children conceived by assisted conception technologies, and the traceability of the culture media really is paramount. So in conclusion, which culture media is best? I don't think anyone can answer that. So if you ask us that on the Q&A, I don't think we know. When I have asked embryologists in my travels how they decided which culture media to use, it has been very varied, their response. But I think people normally stick to what they're used to. I don't think it's a good idea to keep changing culture media, but we can talk about that. As I've shown you, there's not enough studies in the human, and you need to think about the optimum pH for the culture media in your lab. There's a lot of debate about whether you should do this, but I believe most culture medias do say you should check the pH in your lab, and it does depend on air quality. You have to think about the number of embryologists and what type of incubator you're using, and the levels of O2 obviously has been a real hot topic as well, and there are many other variables. So I'm going to stop there. Sorry about that glitch. I have no idea what's happening there. It's like an alien had taken over my computer. So just to summarize, we've had 4 webinars in this series. The first 3 are now on YouTube. This one will be on YouTube later today. And then over the next few days, if you've already emailed us, I have your email. I will send you the link to the YouTube video, but it will be on our YouTube channel, the Embryology and PGD Academy. We have a Facebook group where we will post information about our workshops and any other work we do, and when the modules become live on our certificate. And so just search Embryology and PGD Academy on Facebook. I have 2 Facebook accounts. I'd much prefer you to use the Joyce Harper Work. It has this picture of me here. and this lovely picture of the pioneers of PGT, including Robert Winston, Alan Handyside, and Elena Contangiani, who did the first ever PCR for PGT, and Jacques Cohen and others. And on social media, all of my social media, I'm at Prof. Joyce Harper, and Alpesh on Twitter is at Alpesh Doshi IVF, and on Instagram he's at adoshi2. So if you want to know any more about the certificate, please go to embryologycertificate.com. And if you want to know more about workshops or register for a workshop when we have the date, it will be on embryologyandpgdacademy.com. And any feedback about this webinar or anything we do, then please email us ivf.pgd.academy@gmail.com. Now, Alpesh, I know, has just raised his hand. I've just got to change Alpesh to promote him to a panelist, which I'm doing. Oh. So, Alpesh, hi.
Hello, hi, how are you?
I'm good, I'm good. I'm having this very weird, can you hear this buzzing? I don't know what it is.
Yeah, I can hear that.
Okay, I'm going to mute myself and hopefully it will stop. So let's do the poll first. I have no idea what that is. It's never happened before. I love your background, Alpesh. Very good. We have almost 100 people on today, Alpesh.
Brilliant.
Which is excellent. So let's start with the poll. I'm going to launch the poll. It's definitely something to do with Zoom, because when I click certain things, so you should be able to see the poll now. Which type of culture media do you use? Single step, sequential, not sure, or you do not work in an IVF lab? So if you can just fill those in there as we go along, I'll give you a few more minutes. We're at 73%. So far, we've got the majority of people using single step. So 78% using single step, 22% using sequential. So that's really interesting. And now the next question was about quality management system operational in your lab. Do you use fully quality management system, 45% fully and 45% partially? So that's really interesting. So, right, I'm just going to end that poll now. So thank you. I'll just share that. You should be able to see that now, hopefully. So the majority of people, vast majority, using single step, probably because of time lapse. And then quality management, about half and half, fully. Great to see so many of you fully. I mean, quality management is a huge amount of work, but I'm sure Alpesh feels the same. It's really important to think about these things that are covered in quality management and auditing to really improve the service that you offer in your IVF lab. I'm sure Alpesh will agree, it's very easy to do embryology, but it's very easy to do embryology poorly without really taking everything into consideration. So I'm going to stop showing. The noise seems to have disappeared. I don't know why. Right, so let me just close this. Oh, it's come back again. Right, so yeah, I think it's definitely something to do with Zoom. I don't know what it's doing. Oh, it's gone again. It's gone again now. Right, Alpesh, we're going to go to the Q&A now.
Brilliant.
So Alpesh, while we were waiting to come on, we had everyone tell us where they were from. We had wonderful countries all over. So the first few about this. So the first question about culture media, is it best to equilibrate in dual gas incubator or triple gas?
So that's a very good question. And certainly as a practice, what we used to do many years ago, even when this reduced oxygen concept came in, we were still using dual gas incubators to equilibrate the dishes. Now, we know that obviously when we move the eggs and embryos to a fresh dish, there may be that oxygen shock. if you are using 20% atmospheric oxygen in your culture system, i.e. your dual gas. So now I generally tend to recommend that if you have the provision of a tri-gas incubator to equilibrate your dishes, that is the best thing to do to prevent those temporary shocks to the embryos, especially if you're using a sequential system. So you can imagine that if you're using a sequential system, if you move from your primary medium to their secondary medium, you will be subjecting the embryos from 5% to 20% oxygen shock. And then ultimately it will still go back to 5%. So if you have the capacity of another tri-gas incubator, equilibrate your dishes in a tri-gas incubator.
Excellent. I've just forgot, there's a couple on the chat, so I'm just going back to the chat. One of them says, I'm going to control the nitrogen level. Oh, because the clinic is closed. Right, we're just talking about the clinic being shut. There was another question here. Someone's asked us about doing some ICSI workshops, and people want us to come to Romania and India, Alpesh. We also have someone from Hisam from Sydney. Welcome. So let me just check. There was so the question on here: in the light of the current COVID-19 outbreak, many of us have media stored in our labs, which will now get wasted if their expiry date is near. Is there any way in which we can salvage these?
No, you can't. I think ultimately, if your media is in date, don't worry about discarding it just because we're in the pandemic. If obviously it's an opened bottle, you have to discard it. But if it's a fresh new bottle in its boxes and yet still in date, you don't need to discard it. But if it's past its shelf life, then you have to discard it. That's just the basic embryology practice, and it's nothing to do with the COVID.
Right. Someone said, Ahmed said, if we add procedure, what is minimum time to incubation? I'm not quite sure. That doesn't make sense.
Does he mean what is the equilibration time? So I may just answer that if that's what he means. So that's not a very straightforward answer, unfortunately, that what is the minimum number of hours to equilibrate your dishes. It could be anything from 4 hours to 8 hours. And it depends on the surface area of your dishes over which the gassing has to happen. So you can imagine that if you're using an embryoscope slide, the micro wells have got very little surface area, and it takes much longer time to equilibrate to the correct pH in an embryo slide compared to a 4-well dish, which takes just 4 hours. So the dynamics are not straightforward. There is not a single answer that fits all. You have to be, you have to consult the manufacturer of your dishes, and there has to be validated evidence to suggest what is the minimum number of hours it needs to attain the right pH.
Brilliant. Is it double buffered media an option for pH?
When you say double buffered media, usually culture media only has bicarbonate buffer. If it has HEPEs, I know that in the olden days, there used to be some culture media that had a bit of HEPEs. Most of the newer culture media do not have HEPEs if it has to be subjected to carbon dioxide. So my advice is that if you are using a CO2 incubator, which you would do for culture, try and avoid HEPEs in the culture system. You only need bicarbonate buffer for anything that uses CO2.
Okay, the media like HEPEs or GMOPs is tight capped, and for FERT, cleave, blast, loose capped, is to maintain the pH and CO2?
So anything that has bicarbonate media, in a gas phase of carbon dioxide, you have to have it loosely kept because you want that CO2 to equilibrate the media, because the bicarbonate is not going to work in the absence of carbon dioxide. Anything that has HEPEs, you have to keep it tightly sealed because HEPEs is a buffer in ambient conditions without CO2. So you cannot subject any HEPES media to carbon dioxide. And if you do, that will drastically alter its pH. You will see that the medium will go from pink to yellow if you subject a HEPES buffered media to carbon dioxide.
Excellent. The recommendation of using UVC for disinfection and sterilization for air and surface. Any idea about the formulas for calculating the necessary amount of UV waves?
So generally, UV ultraviolet rays is only used in a lab where there is, you know, in the class 2 flow cabinets. So usually we do not use UV as a method of sterilization. You know, we have disinfectants nowadays that we use in the laboratory. So I would suggest that for your routine cleaning, you just use a disinfectant, and certainly in the light of COVID, do use a disinfectant that is active against the viruses, including COVID. But as a good practice, if your class 2 cabinet or your laminar flow cabinet does have a UV lamp built in, it would be good practice that every night, if you program it to go on at midnight at night with all your consumables in there, you may want to put your plasticware in there, you may want to put your pipette holders, etc., etc. All of this can be in your flow hood in the morning and at night. It would automatically get sterilized because you've got a programmed UV lamp coming on. But please be mindful that you cannot turn on UV in the presence of people working there. You cannot have embryologists in the lab when UV is on, and you can certainly not have embryos outside when the UV is on. So the UV can only go on at night when all the embryos are in the incubators and there is no staff working.
I found out what my problem was and the noise. I have a new external keyboard, which I left in the other room, and one of my annoying children has been tapping it.
Oh.
Oh my goodness. So I did say at the beginning about the trouble with working at home is working with children. And it's the same one, Alpesh, that caused the problem last time. So we can laugh now that I figured out why I was getting all this noise. He was there playing with the keyboard. So it could have been worse. I only just realized. All right, let's carry on. I've told him off. We use sequential media, SAGE, and we add 5% HSA. Do you believe that we should avoid adding this extra HSA? Now, this is a good question because yeah.
So you have to, so even with SAGE, they have quite a few media in their suite. Some of them have HSA added and some of them do not have HSA added. So you can only add HSA to media that does not have HSA added from the factory. Please do not try and supplement additional HSA into medium that already has HSA, because you know what? Then you become a manufacturer. And as per European cells and tissue directives, you are not allowed to manufacture media. The moment you start adding HSA, you are changing the formulation of that media. You would be changing the osmolarity, etc., etc. So if I was the company manufacturing the media and you came to me and said, I have a problem with my media, oh, but I do this, this, and this to it before I use it, I would turn around and tell you, well, you're using it off-label. So you want to make sure that please follow manufacturers' guidelines and do not take these things in your own hand.
I think that was the biggest thing we had from the Culture Media Working Group. The company said to us that people have to follow the instructions. They can't think, oh, well, the other culture media, I didn't do that. Okay, the culture media are very different and they've been optimized with those methods that they've given you. And then that's, you know, to be totally conforming, that's what we should do. This is one about aligning the manipulator. Any tips for aligning the manipulator? It's a good question.
Are we talking about culture media?
No, it's troubleshooting. So it could be anything. Sorry. Yeah.
Okay. Okay. So usually aligning the manipulator, it depends on what micromanipulator system you have. So I work with an RI manipulator, and it's a very straightforward manipulator. Usually the RI manipulator has got an objective. I mean, it's not the manipulator, but it's the microscope with the spacer. in order to align the manipulator with the pipettes to begin with, what really helps is to have a spacer there, which allows you to keep your pipettes at a distance without contaminating them before you start the procedure. So try asking your microscope company or your micromanipulator company if they can provide you with an objective, which is called the spacer.
Brilliant. There's one on the chat. Please put all the questions in the Q&A so I don't miss any. This one was on the chat. Do you recommend equilibrating media in tubes and making the dishes on the day of the procedure, or making the dishes with cold media and cold oil and incubating them prior to use?
I always suggest that equilibration has to be done overnight, especially if it's bicarbonate buffered media, because as I said earlier, we do not know whether your type of dish and whether your type of media needs 4 hours, 6 hours, or 8 hours to equilibrate to the correct pH. So the safest thing to do, and I'm sure all manufacturers recommend that, is overnight equilibration in a gas phase of CO2 and oxygen.
Okay. $1 million question from Annabella. How do you choose your culture media? I've had this discussion so many times. I don't think any of us know, but Alpesh, what would you say?
I think the simple answer is any embryologist or any lab director should always make sure that they have got access to two culture systems, especially if one of them starts faltering. You want to be able to move to another one which you have validated in your own lab. And I know this is actually not kind of recommended, but the way I used to do it in the olden days is we used to do sibling oocytes in 2 different culture media to see what is the performance of these embryos in the 2 different culture media. So I'm still a proponent of that, and I'm not saying that we're experimenting, but we're trying to get prepared to what other culture system would we bring in our lab if product A doesn't work. But again, as Joyce said, what we want to make sure is that there is no one magic media. I have known product A working beautifully in one lab and not working in the other, and product B doesn't work. So I think there's so many factors here that give you results, and not necessarily just the media. It could be the stimulation, it could be your oxygen tension, it could be your VOCs, it could be your plastic wear. There's so many factors which need to be controlled, and ultimately everything will be boiled down to the media, because that's the obvious thing that people start blaming.
You're absolutely right, Alpesh. This is something we really have to stress. And this is why the key performance indicators is so essential. And when I was an embryologist, every month, I think you said this last week or the week before, every week I checked for every person, including the doctors, depending when they did the egg collection, did they do the transfer, what was their fertilization rate, I kept for live birth rate. But it's a much more involved. You have to think about every batch of culture media. It needs to be continuously monitored. Think about your plastic wear, everything has to be monitored. So in module 4, we will cover that. Everything's covered by the speakers to think about how you can have the most efficient lab. Right, the next question, would you like any advice as to why we may be getting bubbles in our culture drops?
So bubbles form when there is respiration. And of course, you know, there's gases being formed, etc. in the culture system. Even when you have embryos, there's gases being formed. So usually in the embryoscope slide, if you're talking about time lapse, usually the bubbles form because you might be using a cold oil. So try warming the oil a bit and then putting warm oil over the culture media and then equilibrating that overnight. That certainly reduces the amount of bubbles. But if you're talking about kind of 4-well dishes, bubbles do form. Certainly when, as I said, when the cold oil is warmed up, you do get some bubbles forming, but you easily remove them before you put your eggs and embryos in there. Or even if your eggs and embryos are in there, you need to try and make sure that your bubbles are removed to the surface and are not in the drop where they're not in direct vicinity to the eggs and embryos, because if the embryos stick to the air bubble, it can dehydrate.
Brilliant. How do you change a culture media? So you make a switch from one to the other. I think that was sort of covered in the previous question. What about rescue ICSI?
It's not allowed in the UK. Rescue ICSI is not allowed because there may well be a sperm in that egg. Okay, so the reason why rescue ICSI is not allowed is you could be introducing another sperm. There may well be a sperm in there, and the fault could have been the eggs for not fertilizing, not necessarily the sperm. So just because you haven't seen fertilization, that doesn't necessarily mean that there is no sperm in the egg. There may well be a sperm in the egg, which has probably not decondensed as chromosomes, or the egg may have not been activated. So by introducing another sperm, you can't do that. Certainly it's not advisable to do that. And I know that there's been babies born from rescue ICSI from failed FERT IVFs, but in the UK we're not allowed to do that as per regulatory standards.
Thank you. So this is about oil. So oil has caused some problems over the years. There was a batch of oil that was affecting fertilization and live birth rate many years ago. Do you suggest to wash the mineral oil with media? If yes, how do you do it?
So in the olden days, that's a good question. In the olden days, we used to wash oil because the fraction of contaminants or water soluble contaminants were quite high. But now the oil is really good. But the answer to your question is that a lot of the oil that we now get, which is commercially available from culture media companies, is already pre-washed to remove the contaminants. So ask your manufacturer of your culture media that, is their oil pre-washed? My recommendation is that if the oil is not pre-washed, try and switch suppliers of your oil company to an oil that is pre-washed. You do not want to start, it's a very expensive exercise to try and layer oil over culture media and then washing it. And again, it's just not necessary in this day and age.
We definitely want the embryologist's life to be easier than in the old days.
Yeah.
So this is a tough one. What study to make if you want to compare two culture media and decide if a new media is better than the one that is used already? And I think for that, it has to be a big study. It's a randomized controlled trial, ideally multi-center. It's something that a single clinic can't look on their own. We do need IVF clinics to do more research. That there are people that are doing these studies. Daniel Bryson's doing a lot of work on big studies to look at this. Thore Hardston's done some work on these sorts of things in the past. So I think for an individual lab, it's not possible, but hopefully some of the big clinics. But if you get asked to take part in a study, I really recommend doing this.
What I want to add, sorry, to that, Joyce, is that, look, I don't want attendees to feel that there is a big difference in culture medias that are available commercially. They are, trust me, they're all as good as each other. It's very important to firstly ask yourself as a lab manager that, am I doing everything as per the protocol that is required by the culture media company? Because trust me, there's so many labs I have been, and they say, oh, I'm using this media or that media. Oh, but that doesn't work for me. But when you start looking at their culture system, the manufacturer may recommend 6% carbon dioxide and they use 5%, or they're not using a tri-gas incubator. So it's all those parameters to control first before you start raising a question on the culture media. The media, let me tell you, are all plus minus here and there.
Yeah, I totally agree. And as you said at the beginning, it depends. You can have one culture media and different labs not working in one and working in the other. So it's good that we've got a range to pick from, but I admit it's hard for the embryologists, but try things out. As part of everyday QC, how to measure the CO2 level of a benchtop incubator, for it is ready-made for use using a pre-mixed gas?
I understand that question. It's a very good question. So whoever's asked that, it's not very easy to be QCing your benchtops when it comes to the CO2 CO2 levels. What I would suggest you do is when you get a new batch of cylinders of mixed gas, you try and directly connect that to a gas analyzer that you may already have in your lab and ascertain what the CO2 level of that pre-mixed gas is and what the O2 level is. That's what I do in my lab. Every time I get a new batch of cylinders, I test the concentrations out with the gas analyzer. And if the levels are fine, then I don't do independent QC test for CO2 or oxygen in benchtop incubators. I would just test the temperatures till I return new cylinders or till I connect new cylinders to the system.
Excellent. My most trouble in the lab is striking oocyte cumulus complexes to plastic pipette tips and also oocytes to plastic stripper tips. How to handle it?
Sorry, I don't understand that. Does he mean that they stick to the plastic or strike? I don't understand.
It must be sticking at there. It says or maybe it says sticking. Yeah. So my most trouble in the lab is sticking. Sticking oocyte cumulus complexes to plastic tip. Yeah, sticking of oocytes of embryos and yeah.
Okay. What I would suggest you do is, this is a known issue. If you're using a polycarbonate capillary, you will get a bit of sticking because of there's a charge on the pipettes. So that there will be a positive or negative charge. I forgot what it is, but you may get that whereby the egg or embryo sticks to either an air bubble which is attracted to the pipette by charge. So this is a problem with your polycarbonate capillaries. With glass capillaries, you don't get that much of a problem, but of course, glass capillaries, the problem is that it can break. So one way to counter that problem or kind of get over that problem of the polycarbonate capillaries is, before you pick up the eggs or the embryos, make sure you wash your capillary 2-3 times in and out with culture media so that you get rid of any of that charge and any of those air bubbles. So without going near the egg or embryo first, go in an area where there's just culture media, rinse your capillary in and out 2-3 times with culture media, and then you should see that you will not get that problem.
Brilliant. So Suhar asked about the academy. Are all the modules presented as a video, or do you have reading materials? So yes, everything has a some text. Sometimes it's pretty much a summary of the talk. Sometimes it may be something to add to the talk. I've written some of them. I might have added a few things. You have a list of references, and then you have the video, and then you have 3 multiple choice questions, which are very similar to the ones in the exam, or even the same as the ones in the exam, which you can just do for practice. So that's how every talk is. And you can go back and watch the talk as many times as you like. It's all very open. Right, thank you. Right, what important gross factors and the quantities? Does it mean growth
factors? Yeah, growth factors.
Growth factors. That's growth.
So at the moment, there's still a lot of research being done on growth factors and culture media. Growth factors should not be taken lightly because they can actually affect the embryo in an uncontrolled manner. You know, you may start growing tumors and everything in embryos, because if there are certain growth factors that are not regulated, then you may be causing cancers, etc. So this is something which is a huge concern. We should not be taking growth factors lightly. It needs a lot more animal studies before growth factors such as insulin type growth factor is introduced into culture media. There are some more milder growth factors that some of the media has, like GCSF, or granulocyte stimulating colony growth factor, GCSF, which is present in some commercial medias, but I don't use a media with growth factors. I use SAGE media in my lab, and it works really well. It's certainly got hyaluronic acid, which is the macromolecule, and it works well. So the answer is, At the moment, I don't think we have enough evidence about growth factors. Certainly not enough evidence about insulin type growth factors.
So we had a lot of discussions about this at the Escher Culture Media Working Group, and growth factors are complicated. They are definitely involved in early development, but they're like the amino acids. It's about a ratio, and we can't work this out for the human. Yves Menezes said to me many years ago, I said to him, I'm worried about these growth factors, and I'm worried about something like large calf syndrome in the human, large human syndrome. And he said that that our 5 days in culture, I mean, he said this long before this was long before John Jamoline's paper. He said that 5 days in culture could affect that person forever. So if we mess around with early development and add the wrong growth factor, we could cause irreparable effect on the person we generate. So we have to be very, very careful. So my worry when we started the Escher Culture Media Working Group was that we were going to see more companies adding growth factors, because it sounds great, we're adding a growth factor, but we have to tread very, very carefully, really.
And some of these growth factors actually control cell cycle checkpoints.
Absolutely,
well. So,
absolutely. They're very strong things in development. We've got to be very careful. Right, according to your experience, what is big difference between Global, Total and Sage, and which is best?
So it would be wrong of us to talk about commercial names and criticize one over the other. So I think it's best that we do not enter into that dialogue of using commercial names on the webinar. As I said earlier, it is how you use the media and not necessarily the media itself. I have used both the products that you have just mentioned. I have a slight preference of one over the other, but again, that's purely because of maybe how I'm using it and not necessarily due to the product.
I think that my take-home message would be that all the culture media are equally good until we prove otherwise. As I said in the study with Daniel, we did show a difference between the live birth rate, and we did try to take in other factors, but it's not that simple. We never named any of the media, by the way, and it's not that simple because, as you said, one media might give a fabulous result in Germany in one lab, and in Australia in a different lab it doesn't work so well. There's so many variables, so many variables. I think it's impossible to say. This question about sequential versus single step, which is the best? Which do you prefer? I would put it in the same category.
I was a proponent 10 years ago with sequential media, totally converted by the Gardner's theory, etc. Then I trialed the single step, and simply because I wanted my life to be much simpler or easier, especially with uninterrupted culture, etc. Now it seems to be the norm that most labs, and as you've seen from the poll, most labs are using single step media in an uninterrupted way. So I don't think there's a right or wrong. A sequential would work equally well, but with sequential media, you have got so many more steps and effort from an embryologist that goes in at changing the embryos, etc., etc. You have to move the embryos on day 3 to a fresh dish of culture media, and we know that the moment you subject the embryos out of the incubator, you are creating a bit of a shock there. So why do that? My preference is single step because it works, and there is not all that steps needed of taking the embryos out and risking losing an embryo because or your pipette broke or whatever. Just keep them put.
And the single step now is not the same as the single step I mentioned back 20 years ago. It's different media, totally different media. Which in your opinion about what is your opinion about antibiotics in the media? Are they really necessary?
So I think that the fertilization media, antibiotic is certainly important. So the answer to your question is yes. I think antibiotics are necessary because there are certain infections in semen or even in follicular fluid. Gentamicin is the most common antibiotic that is used in most of the culture medias nowadays. I would be quite worried to use a media without antibiotics in my lab. I appreciate that uterine fluids don't have antibiotics and it could be non-physiological, but there are quite a few bugs in semen, even E. coli, etc. So I would certainly recommend that you do use a media with an antibiotic. And I can assure you that most commercial preparations do have gentamicin in it because it's a broad spectrum antibiotic.
All right, we're about halfway, Alpesh, so we'll speed up a bit because I know Alpesh's got another appointment. What about the electromagnetic pollution for stressing the embryos?
Electromagnetic pollution, what do you mean?
I think it's very difficult to measure, so we'll move on. Someone wants to know which brand of culture dish you use. I think it's again a tricky question.
Again, a tricky one. When you choose your culture dishes, try and go for dishes which, of course, have got all the MEA testing and have got the very high quality control testing done on these products. So I tend to use the Vitrolife range of dishes, and I love them because not only is the testing really extensive, but also the dishes are they are flat base, so they are in direct contact with the heat rather than with some of the dishes they have an air gap at the bottom, so the conduction of heat is not very good. So again, they're all plus minus here and there, whatever works for you works. I prefer to use the Vitrolife dishes because of the extensive quality control that happens and the fact that they are more in tune with thermoconductivity.
Excellent. With single step media, no time lapse, do you change the media on day 3?
No.
No.
What's important, if I may add there, is that's a good question because so if you were using a big box incubator, I would then change it on day 3 because big box incubators do not have VOC filters. If you were using a benchtop, make sure that there is a VOC inline filter. Then you don't need to change the media on day 3. If you're using the time lapse, that automatically has a VOC filter. So it's all about the VOC filter rather than anything else. But if you're in a big box incubator, do make the change on day 3 because your VOCs can be building up in the incubator.
Okay, excellent. Another question about washing the oil, but I think we answered that. And someone said, should we keep the oil at room temperature in a warmer or gas it before we use it?
So if you are culturing embryos, you have to equilibrate the oil in a gas phase, okay? You cannot culture embryos in a bicarbonate media in an incubator with ungassed oil. And that's why when you make your dishes the night before, they have to be complete with the media and the oil. The only time you can use ungassed oil is with HEPES media, not bicarbonate media, only for micromanipulation purposes. So whatever you're using for micromanipulation has to be HEPES buffered, and that's the only time you use warm, ungassed oil.
Okay, there's a question here about CE marking products. Can you share your experience on how to identify the product, that whether the CE mark is fake or genuine? So this one thing about CE marking is that people can go to different countries to get their CE marking. So I don't know whether the CE marking you obtain in England is the same as the CE marking you obtain in India. I don't know. We did start looking into that, of finding out where companies had. But if you're getting media from a big company, it's going to be a genuine CE mark. They can't lie. Yeah, so yeah. I wouldn't get culture media from a small company. I personally wouldn't do that. I don't know if you want to add anything else, but
all the big 5 in culture media companies have got genuine CE marks. Yeah. So if you take comfort in knowing that if you're buying a reputable culture media, it is a genuine CE mark. But as embryologists, we need to make sure that it is CE marked as a medical device. Okay, culture media has to be CE marked as a medical device, which should have the 4 numbers after the CE. That certifies that that's a medical device. Okay, so that is quite important because you can have CE IVD, which is an in vitro diagnostic, which means that you can't be using an in vitro diagnostic media to culture embryos. It has to be CE marked as a medical device.
but all the big ones have done that.
Yes.
So someone said for dry incubators, single step or sequential?
Doesn't matter.
Doesn't matter.
It doesn't matter. You can use either. When you say dry, I presume they mean non-humidified.
Yeah, yeah.
Yeah, it doesn't.
Emad said,
as long as they use oil. In a dry incubator, it is a must to use oil. Otherwise, the osmolarity can change.
Okay. Someone said, Emad said, what's missing in the culture media till now? We don't know. We don't know. For a normal incubator culture, someone else said single step or sequential. So it's how you want to do it. Pre-mixed gas or not?
Pre-mixed gas or not, it depends on the incubator. So in a time-lapse system such as the EmbryoScope, it does not work on a pre-mixed gas. It needs individual gases. So you have to connect nitrogen, you have to connect carbon dioxide, and it does its own gas mixing. So there is no yes or no here. It depends on the incubator you use, because you may be confined to using only a certain type of gas, depending on the incubator you have.
Yeah. This is a good question, and we've talked about many times. How many embryologists for how many cycles?
Okay, it's very clear. I'll say what the UK kind of norm is. Because of the amount of administrative work that goes on in UK labs with regards to cycle reporting and HFEA and so on, generally the equation we have is about 150 cycles per embryologist.
Perfect. Okay. Thank you, Mila. Mila said that they're a big fan from Malaysia. Right. If there are some fertility centers who use the big box incubators, what is your opinion on that compared to the benchtop incubators, and even better compared to the time-lapse incubator? Can I just say, when I was in embryology, we had the big box, and all this stuff is quite new for me. But with the time lapse, everyone thinks that now we have these shiny, amazing labs, all high tech, automated, etc. I don't know if we're ever going to change the live birth rate. I think we're at the live birth rate globally has hardly changed at all for all these years. I just don't think we can get 80%, 90, 100% pregnancy rate. There's too many factors with women getting pregnant, the quality of the eggs, the uterus, everything. But anyway, do you think it makes a difference?
Can I just add that in terms of the incubators that you've just mentioned, I would not recommend in this day and age that you culture embryos in a big box incubator.
Yes,
and the reason being that every time you open a door of a big box incubator, you're lowering the temperature and pH, which takes 20 to 25 minutes to get back to normal conditions, purely based on the sheer volume of that incubator. So for nothing else, but just the fact that it takes a long time to re-equilibrate back to the ideal physiological conditions. For that purpose, I would not use a big box incubator to culture human eggs and embryos. My recommendation is that you go benchtop or you go time-lapse.
I totally agree. Right, a few questions about pH testing. What is your opinion on the pH testing? How often should we do it? And can you suggest a pH meter? What's optimal? Lots of questions about pH, but.
Okay, so I think I will answer most of these questions by just saying a bit of a spiel about pH versus CO2. So that will probably answer a lot of the questions that you have there. Now, it is ideal and gold standard to be testing pH and not carbon dioxide. Okay, we all know that. But it's not easy to test pH, because the moment you take the dish out of an incubator and put it on your bench, it's losing pH. And because the scale at which we're measuring is so minute that a 0.1 of a pH scale can alter things, you need a very robust pH test if you are going to be measuring pH. So I'll be very honest with you. In my lab, I do not test pH. The only time I test pH is when I'm bringing a new culture system in. So there is a good practice and there is a practical practice. The good practice is that, because it was a question, when should you be testing pH? The good practice is every time you change culture media, And every time you change a batch of culture media, because you can have pH shifts even within the factory setting. So the ideal thing to do is every time you change a batch of culture media or you introduce a new batch of culture media, test the pH. But practically this is not always possible, and hence we measure the CO2 levels in our incubator because that inversely affects the pH. So pH meters are a thing of the past. Certainly in embryology labs, we should not be using pH meters because they are not accurate enough for our application. If you are going to be testing pH, you will have to invest in a lot of expensive kit, something known as the blood gas analyzer. That accurately gives you what the pH is in microdrops. So what I do in my lab, every year I get in fact, I do this twice a year. I get an external company with a blood gas analyzer to come in because I can't afford to buy it. It's quite expensive. I equilibrate tubes overnight in all the different incubators, including my time lapse. He would come in for about 3 hours in the morning and we would test the pHs of all the media. That is my validation.
Perfect. That answers everything. Suha asked about the essay in the academy. This is part of the exit. So Suha, we will discuss this when you finish all the modules. We'll discuss this in detail. Group or single culture? And if you do group, how many embryos together?
Good question. If I'm culturing in a benchtop incubator, then I will do group culture in 4 well dishes and up to 4 or 5 embryos in each well. But obviously, if you're in a time-lapse system, you have to keep them individually.
Yeah. How many days should we use the media once we open the bottle?
The manufacturer suggests it's not necessarily the number of days, but the number of times you open the bottle. Because every time you open the bottle and go in and out, there's a risk of contamination, because you could be introducing contamination into that bottle and you could be changing the osmolarity as well. So some people work in a very busy lab and a 20 ml bottle is used the same day, whereas some people work in a lab whereby a 20 ml bottle would last them a week. So there is no easy answer to it. I think you just need to be cautious for 2 reasons: change in osmolarity and infection control when it comes to culture media. The ideal thing to do is try and use the media. Certainly, I feel once you open the bottle within 3 or 4 days. And again, this is not a magic number and don't quote me for it. I would just think as a good practice guideline, within 3 days, that bottle should be used.
Okay, we're going to whiz through the last one. So what is the pre-incubation time of meat? I think you said you leave it overnight. Yeah.
Yes.
Do you prefer dry incubators or humidified ones? And if it's dry, would you put a dish with water to make a humidified environment?
So we all know the time-lapse system, some of the time-lapse systems are non-humidified. So the EmbryoScope is non-humidified, which is what I use. But at the same time, the benchtop incubators that I use in my lab are humidified. So I think we need a very good study that compares humidified culture versus non-humidified culture to know what the right answer is.
How do you introduce your new LOT in your culture system, and what is the level of oil above the media?
LOT meaning new lot, just new lot.
New lot, yeah. I thought it might be something I didn't know about, yeah.
Nothing much. I literally just keep the certificates of analysis. I check the certificates of analysis that come with all new batches of media. I would just test the CO2 again. As I said, I would not necessarily test the pH or anything in that media. So a new batch gets used as a routine batch as long as we have full traceability of which batch is used when. But we do not have any special measures of quality control before using a new batch, especially with the culture media that is available nowadays. It's so stringently tested. Why do you want to invite so much work in your own lab? We're not making our own culture media, for God's sake.
Yeah, there's another question about humidified or dry, but we've answered that. There's another question about which is better, group culture or single culture, regarding the effects of the bad quality embryos on the good quality. But again, we don't know. So for equilibration of culture media, is it better to equilibrate culture media and oil separately and prepare the dish the following day, or equilibrate the dish with the overlaid culture media?
The overlaid culture.
Yeah. To make embryo transfer, can you recommend to out embryo in media under oil? Is it possible without oil and make embryo transfer quickly?
So good question. I load my catheters literally with media under oil. So I do not move my embryo from media under oil to just media only, just to minimize the oil bubbles or oil on my catheter. I load the catheter straight from the culture dish, which is media or media under oil. So that's how I do it. I know of labs that do not like to have any oil on their outer catheter, so they would move the embryo onto just culture media without oil. I feel there's a more risk of losing temperature, pH, and everything. So I like my conditions to be very, very controlled.
Okay, there's a few people that have written things like which media is better, which media do you use? But I think, you know, we've established that. Someone said in COVID, is it better to put 2 embryos or 1 embryo in drop of culture media? Is it better to change single step media on day 3 in this situation, or is it okay to culture embryos until day 5? We have no idea.
So we have no idea, but one thing we do kind of know, that embryos and eggs do not have the receptors for COVID. At least, you know, the very shallow research that we have so far is saying that eggs and embryos do not have the receptors. So you may not necessarily infect an egg and embryo with the virus.
Yeah. Someone said, how do you take the sample for pH, measure pH in the culture media, in the incubator to analyze it in a blood sample analyzer far away from the lab?
You have to cap your tubes. If it's far, it needs to be done in the lab.
Yeah, it needs to be done.
They need to come to far away from the lab, because you lose the pH.
Okay, someone else asked, what's the maximum time we can use the media once we've gassed it?
So in theory, once your media is gassed, you have to use it within that 5 days, because that's what you're allowed to do, is uninterrupted culture for 5 days.
Yeah. For media preparation, is it safe to mix the media from different batch of media, or I prepare it separately? Well, good quality assurance is to keep it separate, and you need to know what person went in what media.
That's correct.
You can't mix it.
Yeah. If you have 1 or 2 mils remaining from the previous batch, please throw it. Do not use it for the next patient.
Yeah, chuck it. Don't mix them. You should have logs every patient and what you grew them in. There is some info regarding the fact that the plasticware is affected by UV, correct?
I think there is a paper that has suggested that plasticware, the level of styrene and everything can be affected by UV. Generally, I do not sterilize my plasticware in UV overnight, because I just use fresh packs of dishes every evening. I do not reseal mine. So the only thing that gets UV sterilized is the handles, the pipette handles and so on. Usually my plasticware does not get exposed to any UV. Yeah. So to try and just keep on using fresh packs of dishes. Good point.
Okay. Is it possible to use alcohol wipes inside embryology lab or other alcohol?
No alcohol wipes in the lab. Just use wipes that have, such as OOSAFE or something like that, which is tested for embryo toxicity. But do not use 70% ethanol wipes. Okay.
We're going to whiz through the last few. One here about freezing. What can cause degeneration of embryos upon thawing? Zona usually ruptures. I used different batches of thawing media and got the same result. Could it be vitrification media, loading, cryo device?
Yes, it could be the vitrification. It could be the warming. Again, several factors. You need to troubleshoot it quite intricately in stages. If you're starting to see quite a few embryos degenerate post warming, try and see if there is if there's an indication of, is that same embryologist freezing all the embryos? Try and see who's freezing, who's thawing, and try and see if there's a pattern there. I don't think it could necessarily be the media. I know that the obvious thing to feel is that it could be the media, but it may not be the media. It may be the skill of the embryologist. He may be over dehydrating the drop when they're loading the straws.
I think it's training for sure. It sounds like training to me. Another question about group or individual culture, which we've covered. What pipettes are better to use in the culture media preparation, tip or glass?
Personal preference. I prefer to use glass capillaries, but many people prefer to use polycarbonate capillaries. So it depends on what your preference is.
And someone's asked about assessment of the toxicity of the oil.
You should be using a oil which is made by your culture media manufacturer. I wouldn't start undergoing quality control tests of oil in your own lab. And please don't buy oil from just Boots or something like that. I
Yes.
know Boots make paraffin oil, but that is very toxic for embryos. In the olden days, 20 years ago, I know we used to buy mineral oil bottles from Boots and wash it in the lab. No. Now you buy oil from your culture media provider. Full stop.
Yeah, that's a big thing. You can't shortcut your quality in the lab. What is mini IVF and how is it done?
Mini IVF.
Yeah.
Do they mean short exposure? I think what you're talking about is a short insemination time, which is about 4 hours with sperm, and then removing the eggs out of the sperm suspension. There are papers that have suggested that it gives you the same fertilization rates as conventional overnight insemination. In my lab, I do an overnight insemination. I do not have experience with short incubation times with sperm. Of course, the advantage of the short incubation time with sperm is the ROS, is the reactive oxygen species,
yeah,
and potentially the embryo quality.
So someone's asked about measurement of pH per each new batch of media. Is it mandatory, even if the lab is located at sea level? But as you've said there, you don't do it. You'd have these guys in twice a year to check everything.
Not mandatory. The culture media company should be doing their work, which is testing the batch of each media that they manufacture. And that's what they give you in the certificate of analysis. So your job is to check the certificate of analysis that that media has had the correct pH and everything measured.
In your opinion, which is the best technique to load embryos in the ET catheter? Well, in module 1, we've got a video of Alpesh talking through loading the catheter.
Yeah, certainly. I think the 2 air gap method and the embryo column is what I use.
Yeah, and someone's asked on the chat, how long can you keep the embryos in the catheter if it's difficult? I mean, if
it's difficult, the question she's probably asking is, is it better to unload the embryos or just keep the embryos in the catheter? Up to a couple of minutes, I would just wait. I would not unload, because when you unload, the embryos are getting a temperature shock again. So I would just wait there with the catheter till they try and cannulate again and release those embryos. I would just wait for 2 minutes in the room.
I had my twins have IVF and the embryos came back. every embryologist nightmare. Which gas analyzer do you use?
We use a Geotech.
Geotech. I would like to ask quality control, especially measuring temperature in the incubator. Do you measure the temperature directly in the incubator, or temperature in empty dish, or temperature in dish with media?
Temperature in dish with media under oil. Right. Exactly how your embryo culture dish would be.
Yeah.
Not just ambient temperature of the incubator. It has to be, the probe has to sit how an embryo, what the embryo is facing.
Okay. Someone else has asked about which media do you use, sequential or single cell. We've answered that. How do you choose your plasticware?
By looking at what are the quality control measures that have happened on the other side. So is it, are they doing cell counts on blastocysts with the MEA test? Try and understand what is the level of quality control that your plasticware manufacturer is using. And as I said, the other thing to go by is the thermoconductivity, which is a new thing, that whilst we're handling dishes outside, we want to make sure that there is thermoconductivity, heat is maintained in those dishes. And as I said, the new brand of plasticware have got these gaps of airs minimized. So they're all plus minus here and there, whatever works.
Alpesh, do you think you pay for what you get? I mean, some things, yeah, I think you do. I think you've got to realize in this, the more quality assessment a company have done, it costs money, and they'll have to put that cost on the product. Yeah. So I think if you're going to try and cut corners and buy cheap things, then I think you're going to ask for trouble. So I wouldn't do that either.
Not blaming your culture media, but it could be your plasticware.
Yeah. How to determine the source of a contaminated media if you have any in the culture media dishes during the culture days?
So cloudiness in culture media is indicative of an infection. And certainly, again, that's why I would say don't go in and out of the bottle too often, especially not outside on the bench. All culture media has to be opened in a class 2 cabinet. You can't do it on a bench. But the classic signs of infection are, as I said, cloudiness of the media. I have, yeah, it's cloudiness. That's the classic vision to the eye.
So I was asked about maturation media. Have you used it? Which brand?
No, we don't do in vitro maturation.
Yeah. During embryo transfer, the media just flow up, I think it's uncontrollably. What will be the problem, and how often do you transfer with the sandwich method?
Embryo transfer, you mean?
Yeah, embryo transfer, yeah.
I always load my catheter in the sandwich method. 2 air gaps,
yeah,
and the middle gap is the media with the embryos, always.
That's how we used to do it back in the old days. Did you use single-step media and box incubators? Well, we'd say, Albert said, don't use the box incubators. Does your choice of incubator lead to better blastocyst formation? If yes, which incubator? Again, I think we've covered that. In case of emergency, how long can you incubate your culture media before using it?
In a 4-well dish, you want to leave it for 4 to 5 hours. In an embryoscope well, I believe it's 6 hours to 8 hours.
Okay, these are the last few questions. Regarding pregnancy rate, what is the international rate of using fresh semen sample versus testicular sample?
But ejaculated sperm versus...
It just says fresh semen sample. I mean, if you've got fresh, you use fresh. If you've got testicular, use testicular. So, okay, how many days should we use the media once it's open? Alpesh has said that. Do you believe that to divide media in small bottles or use directly from the bottle directly? Because some
That's
companies...
a very good question, because, you know, if we're going to try and minimize infection risk, especially in a small clinic whereby they don't want to waste media, they may want to aliquot the media out. But again, it goes into one of these things that the moment you take the media out of the primary bottle and start aliquoting it, you are subjecting that media to an unknown risk in terms of plastic wear.
Yeah. So now the last...
Yeah.
Sorry, last two questions.
I would say just buy small volumes of bottles. Don't go for a 50 ml bottle if there is a 20 ml bottle available. I know it might be a bit more expensive, but in the long run, you're trying to avoid a lot of problems. Yeah. You don't want to start aliquoting media once you start opening the bottles.
Is the Coda Air device effective?
I don't want to comment.
Yeah, I don't think we should say anything about it. So this is the very last question. And so Rima's got a hand raised. But I just, in this COVID situation, what you said about vitrification with I can never say that, Kitazaku, open system, is it necessary to have separately cryotank for this situation when we have asymptomatic patients? What's the transmission?
That's a very good question. We're debating these guidelines. We're talking about the ESHRE guidelines at the moment. At the moment, the guidelines are suggesting that if it's a known COVID patient, COVID positive, and if you have to do an egg collection, you use a closed system. But if you have someone who's showing symptoms of COVID, you should also try and, as a method of good practice, use a closed system.
Okay, there's one more question.
Although the evidence about cross-transmission of viruses in liquid nitrogen is totally unknown.
Okay, yeah, it's all unknown. Very last question, one more came. You've answered 79, Alpesh. Which semen preparation is good, density gradient or simple wash?
It's meant for different things. So generally, I would use a density gradient of 45 and 90 if it's a good count. But if the sperm is really severely oligospermic, you cannot use a density gradient. You have to do a wash. So it really depends on the concentration of the sperm sample.
Okay, Alpesh, thank you. So Alpesh has got to go. He's got a meeting. I'm going to just finish with the poll so everyone can stay on. Alpesh, I just wanted to thank you so much. These 4 webinars really shows what an amazing embryologist you are, with just such an amazing... I think Imad might want to say something. I'm going to let you... I'll just let you talk, Imad. Do you want to speak? Okay, you should be able to speak now. Okay, I have unmuted. I don't know. It's not. It's your microphone's still on. There's a few people I think want to just okay. Okay, I've allowed a couple of people to speak. Imad, yeah?
Yes, just I want to thank you and Mr. Advish to this huge information. Yeah. And this good information for all the things, for all the 4 webinars. Thank you so much.
Most welcome.
I think that's we've had lots of messages. Alpesh has really done an amazing job. 4 weeks, these 4 weeks, we've really drained him, and we have recorded all of them. They're all on YouTube, and they'll all be on the relevant modules. So Alpesh, I'm going to let you go when I just run the last poll. Thank you so much, Alpesh.
Welcome, Joyce.
Good luck in your meeting, and we'll speak later. Thank you so much. Ahmed, I have unmuted you. You just need to unmute. You can talk now, Ahmed. Okay, I'm just going to run the poll now. So we've got the second poll. Let me just get the second poll up. Okay, that's poll 1. Stop sharing. Now, where's my poll 2 gone? Oh, there it is. There it is. Right. Good. This is just some feedback from you. If you can tell us, did you find this webinar very useful, okay, or not useful? So at the moment, we're on 88% very useful and 11% useful. Thank you so much. So I hope you've enjoyed them. I really hope that soon we can do some more webinars. They've really been great fun, and we've got many, many more topics we'd like to cover. Sorry I keep plugging our academy, but please, you can take a module of your choice for just £100. You'll get a lot more information. You'll get wonderful talks by the world leaders. The certificate will always be open. Module 6 is going to be open probably in a couple of weeks. We're quite along the way now of doing module 6, and modules 5, 7, and 8 will follow. They'll all be live by the summer, and really give you a unique opportunity. So we've been almost 2 hours again. We always seem to be about 2 hours, but we want to answer your questions. So I'm going to end the poll now. So you can see we had 83% said very useful and 19% said useful. So this is really wonderful. So thank you all very much. We'll look forward to seeing you again soon. And I really hope that very soon Alpesh and I can come to all your countries and be involved with some conferences. I want to stress, I really, really love coming to conferences, meeting you all, chatting, giving talks, and it's very hard. One of the things I've really not liked about lockdown is not being able to travel. So we... Hi, Ahmed, yeah. Did you want to say something else? We can hear you if you speak.
No, I just want to thank you for you and Mr. Alpesh for this huge information, and I wish we will meet you again in another webinars soonly.
Yes,
thank you so much.
We will. Alpesh will have a lot of work to do now to get back to his clinic and get it up and running. But we will hopefully do one again towards the end of the year. We'll have to do it probably later in the day because we'll be busy during the day. But it's been really wonderful. And we wish we had time to do some more. But now Alpesh is going back to work. We won't be able to do that. So it was been really wonderful. I love all your questions. We had 83 questions today. So it's amazing. We got through them. So thank you so much. And I'll hopefully see you all again soon. And this will be on YouTube later, hopefully tonight, if you want to see it again. So I'll just say goodbye. Thank you very much. Bye.